How's it going for everyone today? How's it going for everyone today? Let me wait a few minutes to get a few people to come on in. Today we're going to be talking about the basal ganglia and understanding Huntington's disease. And I hope this discussion is going to be useful for you guys. I hope it's going to be very helpful. Let me close my window because I think I hear noise. Hold on one second. Okay. Okie dokie. So I'm going to a few more people get in, then we're going to start. We're going to do a deep dive on the basal ganglia. And this specific disorder is Huntington's disease. So we looked at Parkinson's disease. We did Muhammad Ali, remember, around 24,000 punches to the head, inflammation to his basal ganglia, particularly substantia nigra. That's one of the subnuclei within the basal ganglia. And the death of the dopaminergic neurons in the substantia nigra in that subthalamic nucleus within the basal ganglia dies off. And as a result, you get the symptom presentation of Parkinson's disease. And we went through that mechanism. That Parkinson's disease is the buildup of the intra-conclusion bodies within the cell, the buildup of that molecular debris, that intracellular molecular debris. Impressive, the size of these things and how they damage the structure of the entire neuron. Hey, Hash, how's it going, buddy? Good to see you again, man. So yeah, those Lewy bodies, the inclusion bodies, I believe, is tau proteins that they're made up of. They can get so large they destroy the integrity of the cell. And I was told that fasting is one of the things that decreases the size of that garbage pile. I think it's some way of fasting is a process of reducing that. And so we looked at Muhammad Ali, 24,000 punches to the head. The mechanism is probably through inflammation. Actually, it's probably through inflammation, and I think the inflammation, disruption in the blood-brain barrier, all of those problems eventually result in brain death. And that's how I think Muhammad Ali, the mechanism, whereas in others with Parkinson's disease, is simply the buildup of those tau particles, the intra-inclusion bodies, the Lewy bodies, and somehow the cells in the substantia nigra within the basal ganglia are very vulnerable, although there's buildup of tau throughout other parts of the brain. Some of the parts that continue to have the tau buildup or even more may not be as susceptible to death as the ones in the area of the substantia nigra. So the vulnerability, somehow there's a vulnerability to that process. So that's Parkinson's disease. We went and saw Eric Hernandez, his chronic traumatic inflammation, the CTE, resulted in structural changes in how he perceived things, and it resulted in him doing things that were awful that you wouldn't even think someone like that would do. And his autopsy showed stage 3 chronic traumatic inflammation, stage 3, and that came from football, and he was a professional football player and very accomplished, very accomplished Super Bowl and things like that. And he began doing some of these very awful things to people. And so that's another form of this sort of like brain injury and inflammation. We saw that. And now we're looking at Huntington's disease today, and we'll see um, um, how that works, and we'll do a deep dive on it. Miss Watts, my buddy, how you doing, buddy? How's it going?
Hey, Dr. Reed, it's going good. How you feeling?
I'm okay. I'm okay. It's always a pleasure to see you here on the show. It's always a pleasure. What are your thoughts so far? What are your thoughts? And also, what are your thoughts? How are we doing so far? I'm trying to do this for my students and for my patients and for the family and for the community and things like that. And so I'm trying to keep everyone's interest here. What are your thoughts so far?
I mean, I like that you like break down like the disease processes and how it relates to different parts of the brain. It's very simple, very straight to the point. Um, so I think I like it. I think it's very informative, and I like it. I'm glad that you started doing it. So I appreciate it.
I think it's too technical because we are talking about molecular biology. I mean, we getting down to the nitty gritty.
I mean, some of it can be technical, but then at the same time, you said this is for students, and I think it's important for students to know things at the molecular level so that they can provide efficient care for their patients and know what's going on.
Yeah, that's one of the things I encourage my students to do is pursue their craft till they learn it at the molecular level. Because if you don't understand the process at the molecular level, then you're not really freed from the disease process. You're just trying to do the best you can. I think that looking down there at the molecular level and see where the problem is going wrong there, I think that's the goal of treating patients. At least in my opinion.
No, I agree. Always just the patho is everything. And so I just think that's really important. So
And then isn't it amazing how we apply this at the bedside? I mean, clinically, I mean, we're talking about the receptors and all these and the networks, so it seems like we're able to build a model in our heads and actually use the model to anticipate how the patient will respond. And then remember, we have the classic recommendations, but we seem to have broken away from the recommendations and starting to look at the machinery and say, okay, let's move this, let's move that. And that's because we understand the fine details of the networks and their receptors and how they're functioning, right?
exactly. It gives you a chance when you have like that additional knowledge, it gives you a chance to be really innovative because yes, you have like your standard treatments, but if you're trying to target certain areas of the brain because you know what's going on, then it just gives you a chance to be like innovative and try new things and really doing it from a place where you feel like it's truly the best for your patient because of that knowledge you have.
Yeah, and and I think what we're doing, I think we're so far. What is it like? Have you rounded with other psychiatrists and things like that? What are their makeups like? What is that? What are your thoughts? Is there a difference? Or?
No, it's a difference. I like how you really you really focus on the neurotransmitters and the areas of the brain, where I feel like I've seen other people just really look at like the symptoms, but not necessarily linking the brain.
Psychiatry is so phenomenal and it's so huge. It's the largest branch of medicine. I mean, by far. I mean, psychiatry is everything. It's amazing. It's amazing to even know this stuff at this level. It's amazing. We're ahead of a larger trend in healthcare, and that's the trend to start moving away from labels to mechanisms, right?
Mhm.
So I would prefer to say, hey, all we're treating is a hyperactive mesocortical pathway. Why are we calling these things names? The pathway is hyperactive. Can we slow the pathway down much? Why do we have to call the person all these names? And then the names don't have anything to do with the mechanism that's causing it. And so, it's like two different languages.
Yeah.
You know,
Yeah, and it just takes the people. I feel like it takes away from that bottom patho baseline, like when
yeah,
people just so yeah.
yeah. It sort of allows people to focus on the labels and ignore the mechanism of actions and stuff like that, because that's what we're trying to interrupt here. And then
Exactly.
Here's another thing is that this stuff is so complicated that you can't see this stuff. It's not like you can see things like cardiology. You got a heart, you know what I'm saying? You got a model, and then this stuff is you can't see it, touch it. And sometimes it's just you don't know how much you're projected in the situation. And so you don't know how much you're bringing into the situation when you're describing someone else's experience, So
Yeah.
I'm just for my patients that we're moving that direction. We're going to start label things in terms of the molecular dysfunction as opposed to who they are or their deficiencies. They don't do this, they don't do that. What? Yeah, I'm glad we're done with that stuff. I get we're moving away from those things. It was awful. I mean, it was the best at its time. It's an evolution. I'm so glad we're here. So I'm so glad that you're on my podcast today. And so we're going to talk about Huntington's disease. And we're going to go into the deep dives of the molecular biology of it. And if you have any questions and everything, feel free to answer. And so for my students, I hope they are here because I'm going to require y'all to sign in. I'm going to require y'all to sign in. Okay, we got to find some way to track y'all. Okay. So I should get a lot of questions from these students and stuff and everything. Okay, so do I need anything to start? Okay, so let me go ahead and get started then. So let's see here. I'm going to pull up my notes here. Let's see. Okay. Okay, this is a coronal section, a frontal section of the brain here. All right. Let me get my. Okay, do I need this? Oh, I don't think I need that. Okay. All right. So we're going through disorders of the basal ganglia, and this one is specifically Huntington's disease. So Huntington's disease, HD, is a hereditary progressive brain disorder that causes uncontrolled movements, emotional problems, and cognitive decline. So this hereditary here is an autosomal dominant presentation, right? It's caused by a genetic defects on chromosome 4, and more specifically, one change in this CAG pair causes the actual disorder. So it's just one point, it's like a point mutation, if I can remember, causes all of this. Just one point mutation, right? And it causes the sequence doesn't repeat in the same order. So the one protein that's substituted is different, and then it causes an entire change in the 3D configuration of the protein simply because of that one, and because the 3D configuration isn't the same, because things don't work well. Okay, let's see. I see Dr. Liv came in with a question. Let me just see. Autosomal dominant CAG trinucleotide repeats in the HAT gene on chromosome 4. Anticipation occurs, especially paternal transmission. Thanks, Doc. Appreciate it. Yeah, appreciate it, Doc. So that one small mutation creates all of these different things. Okay. The symptoms typically occur in adulthood and worsen over time, leading to the need for full-time care. There is no cure, right? Okay, so we got that so far. Here, let's see here. Okay, so here, I said here, explain the networking problems with the basal ganglia in Huntington's disease. Okay, and so I wanted to sort of focus on what's the problems with the network with Huntington's disease. In Huntington's disease, the networking problem within the basal ganglia, particularly the imbalance between the direct and indirect pathways. Remember, the basal ganglia has the three pathway structure: direct, indirect, and then hyperdirect. Things the hyperdirect give the cortex full control over the motor system. You can shut everything down like this. Your mind shut everything down. With the indirect, you have the direct and the indirect, direct on, indirect off, and the hyperdirect is just a direct line of turn to inhibit everything. That design is present throughout the entire mammalian species, and it's been preserved throughout evolution. And so, its preservation, the preservation of that structure without evolution over time, and how it's the basis of all mammalian life, makes it a very important structure. So the basal ganglia this networking is a very important networking structure. So when something goes wrong with this network, typically it's very difficult for the animal to survive. This structure is preserved. It's so important. It's preserved everywhere in the function. So we get a problem in it, usually it ends up with the person is unable to function. So these disorders with the basal ganglia are incredibly debilitating. They can become rapid, and the person usually becomes completely dependent upon those around them when this process evolves. So here, the imbalance between the direct and indirect pathways leads to the characteristic motor symptoms. All right. The disease causes a preferential loss of neurons in the indirect pathway, which normally inhibits movements. So in Huntington's disease, the nucleotide change results in dysfunction in the indirect pathway. And the indirect pathway is responsible for inhibiting movement. That's why in Huntington's disease, it's jerky movement because the inhibitor that the inhibited movements isn't really working. So the disease causes a preferential loss of neurons in the indirect pathway, which normally inhibits movement. Okay, this imbalance results in overactivity of the direct pathway and ultimately uncontrolled movement. It's the balance between those two, right? Okay, normal basal ganglia functioning. The basal ganglia are a group of interconnected structures that play a crucial role in movement, cognition, and behavior. Here's where the basal ganglia is located. I should have come up with a slide to show you exactly what the basal ganglia look like, but it's the collection of subthalamic nuclei around here. Okay. They influence movement by modulating activities in the cortical striatal pathways. That's a very important pathway we're going to review next, which involves connections between the cortex and the striatum. This is a major input structure to the basal ganglia. So this loop from your cortex on down there this is the loop from the cortex to the basal ganglia. And the striatal is one part of the basal ganglia, along with several other parts like the putamen, I keep forgetting all the globus pallidum, the putanas, the external pallidum, the internal pallidus, those things. I got to look at them all the time because I can pull them up from memory. Okay, within the striatum, which is a part of the basal ganglia, there are two main pathways, the direct and indirect pathway. So the basal ganglia contains two pathways, the indirect and the direct, and those are located within the striatum. Now remember, there's a nerve bundle that connects the cortex to this striatum, where you have the direct and indirect pathways. Okay, the direct pathway promotes movements by disinhibiting the thalamus, which then excites the cortex, right? The indirect pathway inhibits movement by inhibiting the thalamus. Look at how these pathways influence activity of the thalamus, and the thalamus affects the cortex. These two pathways work in balance to fine-tune movement, initiation, and control, right? So the direct and indirect pathway work together to get you smooth muscle, smooth movement like this, as opposed to like jerking movement, things like that. Okay, so this is the normal function of the basal ganglia. Okay, let's just review. Basal ganglia interconnect the structures, movement, cognition, balance. The influence is connected by a bundle of nerve that goes from the cortex down to the striatal. Inside the striatal, you have two pathways: the direct pathway that promotes disinhibition. The direct pathway disables the cortex, disables the thalamus, excite the cortex. The indirect pathway inhibits the thalamus. Okay. The two pathways working together in balance for smooth movement. Okay. Now, Huntington's disease and the basal ganglia dysfunction. Huntington's disease is a genetic disorder that causes degenerations of the nerves in the basal ganglia, particularly in the striatum, where the two pathways are located, right? So you see Huntington's disease has a predilection for the pathway in the two, for the striatum, but the two pathways are located in the striatum. This degeneration primarily affects the indirect pathway, leading to the loss of inhibitory control over movement. So you're not able to shut off the movements. So this degeneration primarily affects the indirect pathway, leading to loss of inhibitory control of movement. And that's the issue we're dealing with in Huntington's disease. It's sort of like the uncontrolled movement. Uncontrolled movement. Okay, let's see here. As a result, the direct pathway becomes overacting, leading to excessive and uncontrolled movement that we call chorea, right? You getting it? Okay. The imbalance between the pathways also contribute to other motor symptoms like rigidity and slowness of movement, right? If those two are not working well. Beyond motor symptoms, altered basal ganglia connectivity also linked to apathy and other psychiatric disturbances. Yeah, apathy. Yeah, because if your basal ganglia isn't functioning, let's say it's hypodopaminergic, then nothing moves. And so you sit there in the patients sit there in a state where they can't move, but they look like they're apathetic, but they feel normal. And it could because of a movement disorder. All right, but we'll get to that later. Okay, so specific changes in the basal ganglia. Okay, loss of GABAergic neurons, dysfunction of subthalamic neurons. Okay, altered cortical basal connectivity. I'm sure that the wiring would be wouldn't be, there'll be a problem with the wiring in this situation. Okay, let me go here next. Here, okay, the corticostriatal pathways. Okay, this is that nerve bundle that connects the cortex with the striatum, and then we have the direct and the indirect pathways inside the striatum right there. Okay, the corticostriatal pathways are neural pathways connecting the cerebral cortex to the striatum. So that's how that thought get down there to the basal ganglia for movement, right? A key part of the basal ganglia. Okay, look, these pathways are crucial for various functions, including motor control, right? Sensory processing, cognitive processing, and emotional processing. Remember in the case of, let's say, Parkinson's disease with the inflammation and the injury, I'm sure it impacted these pathways too. And I think that's one of the reasons why Muhammad Ali seemed to look so apathetic and simply didn't have an emotional response. And although the damage involved the deeper structure inside of the basal ganglia, that's primarily motor, but he still had all the other cognitive stuff that should have allowed him to just be able to concentrate. So he not only had the damage to the part of his basal ganglia that interfere with his movements, he also had damage to other structures that significantly impaired his cognition. And now with this corticostriatal pathway, that direct connection from the cortex to the basal ganglia, now damaging that pathway, particularly the one we're talking about, is another damage that can actually lead to the diminishing SORM and lack of awareness of environment and cognitive dysfunctions in the patients. And so it's important to sort of understand this cortical striatal pathway and how it functions, because when you're looking at the patient, you're going to have to consider the person's cognitive processes, whether or not they're processing sensory experiences, whether or not they have access to the range of emotional responses or emotional experiences. All of those things could be damaged with this. And you also see patients with Huntington's disease coming in with those clusters of symptoms also. Okay, so here, they are primarily composed of excitatory glutamatergic activation, right? Activation, which sends signals from the cortex to striatum. I think, I don't think you should send GABA that shut things off, so activation. Here's more details. The structure, the cortical striatal pathways are formed by axons in the cortical pyramid neurons and project to the striatum. Great. Function, motor control, cognitive control, contributing to decision-making, planning, and working memory. Reward and motivation, habit forming. Okay, so I just wanted to go through that cortical striatal pathway because it's very important, because again, the two bundles, the direct and indirect pathways are located there. So here, let me just go through explain the different pathways in the basal ganglia. The direct pathway promotes movement by reducing the inhibitory output from the basal ganglia output structures. The direct pathway promotes movement by reducing inhibitory output from the basal ganglia output structures, allowing the thalamus to excite the motor cortex. Okay? It starts with excitatory signals from the cortex projecting to the striatum, the cortical striatum. Okay. The neurons in the striatum direct pathway expresses dopamine D1 receptors and then send inhibitory signals to the globus pallidus. We have that. Dopamine from the substantia nigra excites D1 receptors. Remember the substantia nigra, that's the location where Muhammad Ali's damage was. The decreased inhibition from the internal globus pallidus and substantia nigra leads to disinhibition of thalamus, allow it to activate the motor cortex and initiate movement. Okay, so the indirect pathway inhibits movement by increasing inhibitory output from the basal ganglia output structure, thereby suppressing thalamic activity and hindering motor cortex excitation. It suppresses thalamic activities and it hinders motor cortex excitation. Okay. Let me show you some of the wiring inside here. Okay, this is a frontal section here, looking at the brain, like a slice here. Okay, this frontal slice here. Now, you can see these structures here that's highlighted. These are all the different type of subnuclei, and together they all make up the basal ganglia. So remember, I discussed the striatum right here, okay, and the striatum, it contains these structures, one structure here, the other here, and then the direct and indirect pathway is in this structure here. And so Huntington's disease, this is where the damage is located. However, remember in Parkinson's disease, the substantia nigra with Muhammad Ali, this is primarily where he did get damage everywhere, but the Parkinson's disease symptoms were damaged specifically here to the substantia nigra. And so these two, the location of pathology for each one of these disorders within the basal ganglia are actually different. And so we need to consider that when we sort of start thinking about a plan on how we're going to treat these patients. But that's the level of similarity in terms of the lesion location that they experience. So here, let me see here. Okay, I had this here for a reason. Okay. Okay, let me see. Okay, here, the Huntington gene mutation. Huntington's disease is a genetic disorder caused by a mutation in the Huntington genes, also known as the HTT gene. This gene provides instructions for making the Huntington protein, whose function is still fully not understood, but is believed to be important in the nerve cells within the brain. The mutant HTT gene leads to a production of faulty Huntington proteins, which is thought to play a key role in the development of Huntington disease. So that repeat, yeah, the gene here, everyone has the Huntington genes, HTT, that's located on chromosome 4, right? The HD mutation, the HD Huntington's disease arises when there is a mutation in the HTT gene. The mutation is typically an expansion of DNA sequence. A string of CAG repeats within the gene. The HTT gene gene provides the instructions for building the Huntington protein, right? Although the exact function of the protein is still being researched, it is not known. Okay. The impact of the mutation here. Okay, great. The mutated HTT gene results in the production of an altered Huntington protein. Yep. The faulty protein can accumulate and disrupt the normal function of the nerve cell, which leads to the symptoms of HDD. Okay, I think that's it for now. I'll be open to any questions. And so let me open to any questions. Let me see here what we have next. Okay, so I have Max here. Hey, Nayab. I see Nayab here. Hello, Nayab. Hey, buddy. Okay. And then Max, how is Huntington's disease genetically inherited? Autosomal dominant, HTT gene, the CAG repeats, the folding pattern of the protein makes it unusual, and you can't use it. Hey, iHash, how's it going, buddy? Can lifestyle changes make a difference in the symptoms? That's tough, because this happened at the molecular level. I would think you would probably have to stay in the best shape you can, have a very restorative routine, have a support system. I think, you're going to need to do a lot with that, and you need to do a lot with that. Okay. And then Doc got it right. Congratulations, Doc. Charles. Hey, Charles. Interesting that Huntington's presents in childhood despite being genetically present at birth. Yeah. Yep. Yeah. However, at each stage of your life, there's a preferential different set of genes that are preferred, used, and activated. So in early childhood life, you have these childhood genes, adolescent, early age. And so the brain is actually a mosaic of all the individual pieces that grow and develop individually, right? At times, patients with developmental issues or genetic disorders, the networks are not repaired the same way, and they're not updated. So you shouldn't have the same proteins in parts of your brain as an adult that you had as a child. If you did a biopsy and you look at them, you have the same proteins that a child would have, then we would look at those areas and say, hey, those areas are underdeveloped because they haven't matured enough to even express the adult proteins that typically manage these issues. So we see that in the form of like learning disabilities and things like that, that the person continues to grow and the patient and the parts that are damaged don't grow. So the person is sort of stuck at that developmental level because all of the network hasn't been updated to the updated skill set. Make sense? Hope it wasn't too complicated. Okay. Hey, T-Jam, how's it going? Autosomal dominant. Hey, Umber, thank you so much. Why does the generation of the indirect basal ganglia pathway chooses career? It chose okay, chorea is just a general name for the type of movement. And so when they say it calls rapid unexpected movement, you can sort of like classify that more as a career for movement and stuff in that situation. But rigidity is also a part of it because it can go from relaxed to rigid. It can go from fast to slow, from slow to fast. Okay. Next, Charles, how's it going, buddy? How do you tailor the clinical approach for a genetically determined disorder like Huntington's disease compared to one with a complex multifactorial condition like schizophrenia? You do the best you can. You do the best you can. If you look at all of medicine and everything, there are certain categories. You're going to see how can you intervene at the molecular level with the medications. At the system level, how do you intervene at the system level with like nursing, with caretakers and things like that, working on organ systems. But then you have other things like environments like homes, communities, and so addressing symptom things at the level of home, like home health care, home assistance. They may need things like detox. They may need nursing homes and things like that. The healthcare system addressing those things. And then finally, another range of services that help the patient sort of maintain their spiritual life, their cognitive functioning, their connections to their families, their connections to their communities and stuff like that. Those are all specific parts of the healthcare system where we try to help use the tools within those areas to deliver the best services we can. And the area that I work, it's not all of those services are not available. So we have to try to do the best we can for these patients. So you end up trying to do the best you can with a medication plan. We end up do the best they can with doing an evaluation to make sure they get a thorough evaluation plan and make sure their treatment goes successfully. We do a good job in helping them get to nursing homes or shelters or group home or things like that. And then I think our society, our healthcare system doesn't have much to help with the holistic part of and help the person maintain their healthiness and stuff like that. So that's a weaker point of our system. So the complex nature of these disorders, we sort of handle them within the different sections in our mental health system. So that's how it works. Okay, and I didn't want to take these lectures too far. It's 37 minutes now. I want to try and keep them next 20 minutes. And I don't think there's anything else I need to do with the basal ganglia. I think besides Huntington's disease, I think we're sort of off on this subject. So I think I'll wrap up the discussion on the basal ganglia. If there's anything else, if there's a different disorder that I miss, just let me know, and then I'll see about doing it so that we can. So I want the discussion for the basal ganglia to be complete. So in the next round of students or families, we can put the. So maybe Fahad, you think we can go ahead and put the. Miss Watts, did you have any questions for us? Anything like that? I forgot. Girl, give me some input. What are your thoughts about it? I'm going to the next part of the show. Girl, what are your thoughts? I'm sorry.
No, I didn't have any specific questions, but I thought as far as the rigidity goes with Huntington's, I thought or the lack of rigidity rather, I thought that was because that inhibitory factor is damaged pretty much, and so because you don't have anything controlling it, saying okay, we can't stop, since that part is damaged, there's no control mechanism, so that's why there's a lot of movement going on.
Excellent. Hey, that's the same experience I have when I try to go through that, because I say this structure has been around since the beginning of time. There couldn't have been any dinosaurs. I guess the dinosaurs had the same switching mechanism, direct, indirect, and then it's hyperdirect. But every time I try to go through that process, I end up saying, yeah, and all of that. So I agree with what you're saying, but it makes sense to me, but let me just put it like this. All of those different type of bizarre movements are still considered as uncontrollable movements. So even the rigidity, because they'll be walking and all of a sudden rigid and then fall, boom. So that was sudden rigidity, right? And I'm like, well, how do you get sudden rigidity? You had to get activation of the active direct pathway to turn things on, but you didn't turn things off? Or did you turn off the act? You see what I'm saying?
Mm-hmm.
Well, at least you know it exists and stuff like that. At least you know it exists. Just do the best you can. It's unfortunate that this disease is sort of carried on like that, and it's not really a lot of treatment options besides supportive treatment for the patient.
Yeah.
Mhm. Okay, well, thank you so much for appreciate this. Thank you much for being on the show. Okay,
Thanks for having
okay, and if there's any other questions, I'll talk to you guys later. It's been a pleasure, and read out. Take care, guys.
me.